So as I mentioned before, pepsin has the optimum pH to work with. It's around pH 2. For trypsin, it is a little bit, it's kind of neutral to a little bit basic. It is around 8. So it depends on, depending on the enzyme, each one will have a different optimum pH to work with. So another enzyme that we want to introduce is called kinase. Most of the enzymes, they end with ASE. Whenever you see a scientific term that ends with ASE, then most of the time it is an enzyme. But of course, some of the enzymes, they do not end with ASE, just like what we saw previously. Pepsin and trypsin, they do not end with ASE, but they are still enzymes. So kinase is an enzyme. What does it do? Kinase is an enzyme that will put a phosphate group onto a molecule, onto a substrate. So it's basically, it just grab a phosphate group and then put it to form a covalent bond it with a molecule. In this case, it is a protein. And a lot of time, a protein or enzyme or other molecule, when you get a phosphate group, it can be activated. They can turn on. So you may assume that this process requires phosphorylation, putting on the phosphate group on the molecule. It's like an on-off switch for a molecule. In this example here, an inactive protein can be turned on or activated by putting phosphate group. So in this case, kinase activate this protein. But there are other proteins that works the opposite, meaning that when they get a phosphate group, it can inactivate them. It can turn off. So not all enzymes are needed all the time in the cell. A cell regulates the metabolism by regulating the enzyme activity. If the enzyme is active, then the metabolism will be faster. So the enzyme can be turned on and off. It is important because it is regulated by the gene. The situation is like the gas pedal. When you're driving, you have the option to step on your gas pedal in order to accelerate, or you can release your gas pedal to decelerate. So the gene producing the enzyme can be turned on and off is similar to this idea. You can step on the gas pedal or you can release your gas pedal. And this phosphorylation, putting on phosphate group, will change the shape of the molecule. There's another analogy. It's like stepping on a brick. So I mentioned that this one gene can turn on and off of an enzyme concentration. It's like stepping on the gas pedal, releasing gas pedal. And there's also a brake. You can step on a brake to decelerate, or you can release the brake to accelerate. What is the brake? The brake is the enzyme inhibitor. Enzyme inhibitor is like a brake on the biological system. Now we talk about enzyme inhibition. So we talk about the brake now. So when substrate cannot bind to the active site of the enzyme, then you have inhibition. And you have feedback inhibition, or some people call it negative feedback. That means when you have too much of the product, sometimes the product can bind the active site and block the substrate from going into the active site. In this case, we call it the negative feedback. I can draw it here. You see. To enzyme, and then become product. Product will actually inhibit here, so that the substrate can no longer bind to it. So the simplest way for inhibiting enzyme is for the product to bind to the active site of the enzyme, so that the substrate cannot bind to the active site. When the product is used up, of course, the product will detach from the enzyme active site, and then enzyme can bind to the substrate again to make more product. And then there are other types of enzyme inhibition other than blocking the active site by the product. For example, cyanide can inhibit an essential enzyme. I believe it's cytochrome. Penicillin blocks the active site on the enzyme as well. So all these, they are the enzyme inhibitor to cause biological changes. Usually these are the fatal ones that can kill the bacteria and also kill human being. So enzyme inhibition. So at first, you have the reactant or substrate bind to the enzyme, and then you have successive chemical pathway, and then the end product is F. But if you have too much of the product, what will happen? The product can bind to the another site of the enzyme. In this case, it is not the same active site. It is a modulating site that can cause the conformation changes to the enzyme 1. And then in this case, when the shape of the enzyme change after binding the product F here, then the shape of the enzyme will change. And then the shape of the active site will also change. Then it can no longer, the substrate can no longer bind to the active site. In this case, then the enzyme is being inhibited. So this is what I meant. So the binding of the end product to the enzyme 1 will change the site, the shape of the active site. And in this case, the reactant can no longer bind to the active site because the shape, they're totally different. And in this case, the we call it, it is an example of inhibition. This is just a summary. Cofactor, I briefly mentioned about cofactor. Cofactor usually is an inorganic ion or protein molecule. What happens is that okay, the ion can couple zinc or iron. That's why if you take Centrum, the multivitamin, they usually include all these inorganic ion into that because they know that enzyme require this inorganic ion for cofactor. For non-protein cofactor, we call them coenzyme. So vitamin is an example of coenzyme. Remember, we learned about in the we haven't talked about cellular respiration, but in the next lecture, in class lecture, we'll talk about cellular respiration. Then in that lecture, we'll learn about coenzyme play an important part in producing ATP. Oxidation reduction reaction, or many chemists just simply call it redox reaction or redox reaction. So one way to memorize it is the oil rig. So oxidation is the loss of electron, reduction is the gain of electrons. So it helps you, this mnemonic helps you to memorize the differences between oxidation or reduction. The definition actually extends to oxidation is the loss of electron or hydrogen. Reduction is the gain of electron or hydrogen. So you cannot have one oxidation without reduction. They always occur in pair. Again, it's like yin and yang. You have oxidation, you always have reduction. So they always work in pair. That's why we call it a redox reaction. So this is an example. When oxygen combined with magnesium, then oxygen gains the electron, and then magnesium loses the electron, and then it becomes oxidized. So this is an example a redox reaction. So what it means is this: magnesium plus oxygen become magnesium oxide. Since you have 2 here, you need 2 here and 2 here. So they are talking about this reaction. We can break it down into 2. So this is what it means. For oxygen here, it is becoming reduced. Because oxygen gains electron, then it is reduced. For magnesium, it loses electron. So it is become oxidized. So this is oxidation. This is reduction. When you combine these 2 chemical reaction, you get this overall general equation, the reaction. So it says that oxidation reduction, it can occur when oxygen is not involved. For example, remember NaCl. When you have sodium ion and chloride ion, when they combine together, you form sodium chloride. This is also an example of a redox reaction. Remember I told you the example about getting married. So he has a diamond ring to give it to the woman. So remember, it is a transfer of an electron, complete transfer of electron, which fits into our definition. Loss of electron and gain of electron. So in this case, sodium is oxidized because sodium lost an electron. He gave a diamond ring to the chlorine. Chlorine is reduced because it received 1 electron, so that it becomes chloride ion. So it is reduced. Therefore, it is an example of a redox reaction.